We'll give everybody a couple minutes to sign on. Has anybody taken an incredible vacation or something we should know about?
I wish I had. Have you? You took the twins away. Did you have a good time?
I did. I took them on for everybody who's not from Texas. There's this kind of ghetto little town, but it does have a beach. It's Galveston, and cruises leave from there. You can get in the water. You also might get a brain eating amoeba if you stick your foot in the water. But that is beside the point. Like once a year, I see that headline. I'm like, why'd they go to Galveston? Nice to look at though. And the cruise had water slides and putt putt and an ice skating rink and all of the things. I was super excited because I was like, well, this is great. The twins are captive. Um, but it was still a lot of work. But I knew that they had to stay on the ship. They couldn't wander off too far.
Maybe overboard.
That was Disney. This was Royal Caribbean. Hi, Chris. I'm relieved to see you on. There are some topics that I can introduce and talk about a little bit, and this I was like, oh, I got nothing. It's probably better if I just totally go on mute and off video the entire time.
Sure.
Okay, it looks like we have everybody on, so I will get started. We have some announcements this morning. Welcome to the virtual monthly lab meeting. I have my joke. Organic chemistry is difficult, and those who study it have alkalines of troubles. Or all kinds, I'm sorry, all kinds of troubles. I thought that was a very, very good one. I see some nerds' shoulders shaking. We are a patient advocacy group dedicated to improving the lives of children and families affected by SLC, and we stop at nothing to get treatments into clinic. I think everybody knows we have our symposium coming up. And Laura, would you like to say a couple of quick words about it?
Sure. Hi, everybody. I won't say good morning because I know everybody's on different time schedules. Our symposium for 2025 is going to be in Atlanta. We have a location in Midtown at the Georgia Tech Hotel and Conference Center. The conference will be December 4th for the scientific symposium. And we are having a 2-day conference, so the second day on December 5th will be for families. We'll be at the same location right now as the SYNGAP Research Fund, and we'll have biospecimen collection as part of the Combined Brain Roadshow, and a respite room for families as well. So our website should be live in just a couple of days, and I know Amber will share that with everybody. We do have the scientific registration up right now as well. So any questions, please reach out to me, and we're really excited to be in Atlanta this year.
And Laura, could you put the registration link in the chat?
I will.
Awesome.
Got it.
You guys, Laura does all of this every single year, and she's received a trophy 2 years in a row. And this year, I asked Porsche because of Atlanta, but we'll see if they come through as a thank you gift. Not super optimistic. So I can't emphasize enough, you're burning daylight. If you haven't registered yet, stop burning daylight. and register. It's a great time every single year. And this year will be absolutely no different. We are already ordering our merch. And now, Lindsay Randall from SLC 6A1 Connect Europe is going to give an update on our European conference. And take it away, Linz.
Can I share a screen? How do I do that?
Yes, I need to stop sharing. Now you can.
Share screen. Thank you. Okay. Yeah. Hi everyone. So I'm Lindsay Randall, and I am the founder and CEO of SLC 601 Connect UK, and I am just going to give you a really brief introduction to me and also to the European Alliance and the European Symposium, which we will be hosting in August. So I founded SLC 601 Connect UK, also known as Arthur's Quest, in 2019. And I have two children with SLC 601. You can see in the picture there, Arthur, who's 9, and Eden, who is 5. For those of you that don't know us, they're both pretty challenging, super cute, very sweet kids, but also, flip at a switch of a button like many SLC kids. Arthur is verbal. Eden is not verbal yet, and they both go to special needs school. About a couple of years ago, probably 3-4 years ago now, time flies super quick, more organisations started forming across Europe, and it felt really odd to me that we weren't working together and we were all doing, basically the same work, but like completely individually. And then obviously we have Amber and Connect over in the States, sort of like being our big kind of parent organisation. So we came together, and first and foremost, we decided to host a European symposium. And the reason we decided to do that is because lots of the European families in particular, but also the researchers, can't get all the way to the States. I have been twice. The symposium's amazing and they do a phenomenal job. And it's so insightful and you learn so much. I thought, how can we, we want to increase interest over in the States. We want to enable people to collaborate, to network, to learn from each other, and we want to enable European families to be able to attend and learn and meet each other, which is not always possible when it's hosted over in the States. So we are running it. It's the day before the International League Against Epilepsy Congress. We actually are having a 2-day, so a little bit like Amber did in Florida. We have a family bonding day on the 28th. And then we have the kind of conference day on the 29th, and it will be split into 2 rooms. So we'll have a family conference in one room and a scientific conference in the other room. We'll all come together in the afternoon and we'll all go out and have a reception in the evening. And we're co-hosting, so GABA are having their own conference in the same hotel at the same time. So we'd love to invite as many of you as possible, anyone interested, particularly those who are over in Europe. We have lots of European projects showcasing, but also some of our amazing American project presentations
Listen to
are
this too if you want later.
coming over. And yeah, so the link I'll put in the chat. We've got all the information, and much of the agenda is posted on the Eventbrite page now. And just a quick introduction. So SLC 601 Europe, these are all of our European organisations. As I said, we're growing across Europe, and it made sense to come together. So that is essentially what we've done. We've got a scientific advisory board, again from across Europe and from the States. And we have a list of our contacts from the European Alliance. So it's not a registered entity in itself, but most of the members are registered non-profits, with 3 centres of excellence, which are Sant Pau in Barcelona, Danish Epilepsy at Philadelphia, and also in Subria in Italy. And that is our introduction. Thanks for listening.
Wonderful. Okay, back to my slides. Oh, and Lindsay, if you could put the link to register in the chat.
I sure will. Thank you.
One update from Jacob Tiller, our CSO. We have a number, here, let me read it actually. The biomarker project is underway, and if there are any publication opportunities available in the meantime, he can help write things up, work with you. We have so many journals reaching out to us to publish, and it's very rare that you have talent to help. So please let us know. We do have some new research put out by All Stripes. It essentially said that we have a very high burden, a very high family burden to carry, looking at real world data that was collected through online records. And I will put a link to that research in the chat as well as my wrap up. And for the clinicians on the call, I just wanted to pass on a really cool resource called RareCap. Actually, Sarah Poliquin on this call told me about it. It's a collection of clinical protocols for rare diseases, and there are a few genetic neurodevelopmental disorders already listed. Protocols must be submitted to this database by a clinician. But if you are interested in supporting it, I will put Sarah's contact information in the chat as well as my wrap-up email. I think everybody knows we have 3-year natural history study available, centers of excellence around the US, and available iPSCs in mice. I have it all in a Google Doc now and a really quick PowerPoint that I'll send out afterward. And in my favorite thing to talk about is good things happening to good people. And Dr. Caddy Kang has been promoted to professor. Everybody give a round of golf claps because we could not be more proud of our hero. Thank you so much for everything you do for all of us, Caddy. We are so proud of you.
Thank you.
And she has a son going to college right now.
Study neuroscience. SRC. Oh, no, neuroscience.
And the circle of life continues.
We need the next generation neuroscientist.
If anybody is new on the call, feel free to introduce yourself. I know we have a new intern, Hamilton. Hamilton, would you like to give a couple words?
Hello, everybody. My name's Hamilton Ripkin. I'm a rising sophomore at the University of Notre Dame. I am currently studying neuroscience with a minor in rare disease, which is a super amazing program that they have at my university. I began working with SLC this past semester, and I've loved every minute of it, and I'm just excited to continue working. And if anyone has any opportunities or anything I can help out on, I'd be happy to help. So, hello.
So our interns, Jacob Tiller, our CSO, is currently working with Dr. Greenspan right now and working on the biomarker project and involved in everything. And Hamilton is working with Dr. Rotenberg and Dr. Lee at Harvard right now. And he is sitting across from Chris McGraw's old office. How amazing is that? You need to go take a selfie in his old office just for our little collection of random pictures. I have a nice one of me just sitting in his chair when he's not there. So now, and again, if anybody is looking for an internship, interested in science, we are so happy to accommodate. And now Dr. McGraw, who recently went to Northwestern from Harvard, and he is speaking about something so far over my educational level that I'm not even going to bother an introduction. Chris, take it away.
Okay, thank you. I'm sure it's not over your head at all. As you'll soon see. Let me just share my screen. Can we all see this?
Yep.
Okay, great. Let me just get something up here. Great. Okay. Title of my talk is High Throughput Imaging of GABA Fluorescence as a Functional Assay for Variants in the Neurodevelopmental Gene SLC6A1. I have recently moved to Northwestern after being at Harvard and Boston Children's and MGH for some time with Henry Lee. And as you mentioned, Amber, yeah, you had the picture from my office that you showed me later that showed that, yeah, you'd snuck in at some point without me knowing, which is cool. And yeah, would be great to see another photo by Hamilton maybe in the same place. But without further ado, happy to talk to you guys about this assay that we've developed and put it sort of in the context of our wider work. So the overarching interest of my group is really how do we go about discovering better anti-seizure treatments for all different types of epilepsy and seizure disorders like status. And to me, this really breaks down into two main branch points, one of which is expanding the target diversity of anti-seizure drugs. And you can do that sort of based on informed hunches, or you can do that in more large scale screening operations of compounds or identifying genetic modifiers. And that's one of our areas of interest. The other one is developing better understanding of the mechanisms of specific rare refractory epilepsy conditions or refractory seizure conditions. And that, of course, requires new models of those disorders or of the genes and the proteins involved in those disorders. And of course, having two of these will hopefully synergize to allow us to go further faster. And my group is interested in a lot of different things and a lot of different tools. We use human data, mouse, zebrafish data, as well as cell lines. And the study that we're talking about today is really exclusively just the work that we've developed with a cell-based model of cell-based fluorescence assay for GAT1 function. And this room probably needs no introduction to SLC6A1 related disorders, but it's good to review the basics. It encodes GAT1, GABA transporter 1, primary transporter involved in GABA reuptake in the central nervous system. Shown here is a diagram of its protein architecture with the effects of different variants at different positions on GABA from a recently reported paper, but this just shows you an overview of the spectrum of variants and their location and the level of severity associated with them. Genetics are believed very strongly supported by evidence to be haploinsufficient, largely de novo variants with a relatively high percentage of missense variants, although there's some premature truncating variants too. It's associated with epilepsy in the vast majority, many different types. Of particular interest is the epilepsy with myoclonic astatic seizures, but also other generalized seizures and other seizure types. It's also associated with developmental delay, intellectual disability, and autistic traits. And the mechanism, which has been well elucidated by people in this room, like Caddy King and many others, it appears to involve reduced GABA transport, and perhaps in a large percentage of these variants also is related to a reduction in the surface expression of GAT1, which has the overall effect of also reducing GABA transport. And the treatment for SLC6A1 related disorders, and specifically with seizures, involves conventional ASMs, but many can be refractory. And so other things that have been tried include ketogenic diet. There's been a report of acetazolamide, but there's some concern maybe these are just focused on seizures. They're not going to affect the developmental trajectory or the intellectual disability. And so greater interest is maybe in the effects of disease-modifying treatments, which could include the molecular chaperone for phenylbutyrate, which is in a clinical trial and has been studied by Caddy's group quite extensively. Gene replacement is another possible avenue. And one that hasn't been talked about much is the idea of selective GAT1 agonist. And that was, although I can't report any progress on that front today, that was actually one of the motivating factors that had led us to get into this and to develop an assay based on fluorescence to try to do drug screening for GAT1 agonists. So as I just alluded, that was a major factor in us getting into this. But in addition, there's an ongoing need for the functional validation of SLC6A1 variants. I think when we started this project, there were maybe 100 that have been characterized. I think that's now greater than 200 at this point with respect to variants that have been tested by some functional assay involving GABA uptake. But there's always more. And so having an assay that might have more convenient properties is beneficial. And also just recognizing some of the drawbacks of the tritiated GABA assay for high throughput screening. It does work. I mean, it is high throughput, but there's the extra burden and regulatory issues related to handling radioactivity and disposing of it. And fluorescence assay fits maybe more neatly into some existing high throughput pipelines based on high content imaging. So the objectives of our study were to develop a fluorescence-based assay for human GAT1 function using the genetically encoded IGABA sniffer protein that's been described in the literature, to use that assay to assess the effects of human variants on GAT1 function, optimize this assay to be able to assign pathogenicity in the same way that has been done with tritiated GABA, and then to also do some optimization towards its use for compound screening. So in essence, the assay that we developed has two parts. It's a heterologous assay in HeLa cells that involves a construct expressing human GAT1. It's tethered by a P2A linker to TD tomato. And in addition, the second part is a IGABA sniffer that's been modified to remain cytosolic, so it's not being expressed on the cell surface. It stays within the cytoplasm of the cells, and this is linked by P2A to an MBFP. And in essence, the theory of operation of this assay is that when at the baseline with no exogenous GABA, these cells should have some very low level of IGABA sniffer-related green fluorescence. And in the presence of exogenously applied GABA, we have GABA uptake that's mediated exclusively by the human GAT1 that's being expressed heterologously, and then gives us a good readout based on this change in the cytoplasmic IGABA sniffer fluorescence that indicates a higher amount of GABA. And our approach to this was using HeLa cells, either transient transfection. We also have a stable cell line, 96-well or 384-well plate, using the Image Express micro automated high content fluorescence imaging along with some of its analysis packages. And from that, we derived these estimated GABA transport values. Essentially, as we'll discuss, it's sort of like a steady state level of GABA after there's been some prolonged incubation. And here's a representative example of what this data looks like. It's very similar to what's been published with respect to this GABA sniffer, when at its inception. But we see that in the context of this assay with our combined fluorophores, you can see clearly here that the cells that are co-expressing MBFP, which is linked to the IGABA, as well as the TD tomato, you can see that in the presence of 100 micromolar GABA, there's this increase in that relative amount of IGABA sniffer fluorescence that's indicating uptake of the GABA media by GAT1. And this is what some of the data looks like at the level of an individual site. So we scan multiple sites within the well, and here just showing from over 200 cells, there's a clear shift in the level of the IGABA. And when it's averaged across sites for the well, you can see an even nicer delineation between these two values indicating effects of the GABA uptake. So we validated this first for sort of gold standard loss of function variants involving domain deletions. It had been reported previously that there's an abrogation of GABA uptake when these domains are deleted. So we had gone ahead and deleted this N-terminal CPD cytoplasmic domain. We also deleted one of the extracellular domains and then evaluated the effects also of deleting 2 of the transmembrane domains 6 through 7. And what we see is a very nice effect, particularly at the lowest levels of GABA, where there's, if you look, for example, in the wild type data between no GABA and then just 1 micromolar of GABA, there's this very dramatic increase in the levels of GABA fluorescence for the wild type. A little less dramatic, but even higher for 10. And then you can see that these are pretty reduced for the variant related to the ECD as well as the CPD and the TM6 and 7. And for most of the data, we go on to report it really just as a normalized change, a fractional change over the variant's own vehicle condition. And you can see it even more clearly here that 1 and 10 are really very specific, whereas at 100, there starts to be a higher amount of GABA uptake that is apparently not mediated by GAT1 up to 30%. And so our studies had kept our levels of GABA below 10 for the majority of the assays. We then went on to ask, in the context of human variants, how the IGABA assay would perform. And for this, we took a number of variants, focused on ones that had some report of epilepsy, also the most recurrent variants that were known back in 2020 when we had started the study. And we used 7 variants that were thought to be pathogenic or likely pathogenic, 2 that were believed to be benign or likely benign, and then another 7 that were believed to be of uncertain significance or had conflicting reports. And sure enough, we see very much sort of expected results related to, in relation to data that has been seen with the tritiated GABA uptake assay. Here, we're just showing a series of wild types run from different assays, just showing the level of variation between the wild type controls. Our benign variants seem to have very similar levels of GABA, of this fractional GABA uptake, whereas deletion variants have very little. Our pathogenic variants are reduced. And then amongst the variants of uncertain significance, we see sort of 2 clusters, one which appears to have has significant residual, if not normal, GABA uptake activity, whereas there's some that appear to be strongly reduced. And you can see using a cutoff based on the 95th percent confidence interval from the pathogenic variants that we tested, you can divide these VUSs into 2 different clusters. And so it looks like this could be helpful for variant resolution. And the results appear to be very similar to, for example, Silva et al., a recent paper that had reviewed more than 200 variants using the true data GABA uptake and surface expression data. It seems to be closely aligned with those results, and we'll talk more about that on another slide as well. The assay seems to have a pretty good test-retest reliability, so the same variants run a second time seem to be give highly correlated results with R squared of 0.58. I'm sorry, 0.85. And so that some of you might be wondering, what exactly is the correspondence between IGABA and radioactive treated GABA uptake? Are they really measuring the same thing? In the case of IGABA, we believe this is a steady state intracellular level of GABA. It reflects the I would call it a pseudo steady state after 6 hours of accumulation, when there's no more coming in, there's no more going out. It's just sort of flatline. Whereas with a tritiated GABA uptake assay, typically it's a rate of GABA uptake that's measured over a very short application period, and it's likely it's a different pharmacokinetic sort of measure, and it's not clear maybe what the correspondence is. But what we can say based on published literature, we took the data from this large series that was published in Silva et al. From these variants based on the tritiated GABA assay and compared them to values that we got from the IGABA assay. And indeed, the correlation seems to be quite strong. Let's see. I'm sorry, I see there's a question. Oh, regarding the GAT1 independent uptake at 100 micromolar GABA, do you still see it in untransfected cells? Yeah, maybe we'll talk about that at the end. There's something weird that appears to go on. I don't know if I have a slide about it, but the long and the short of it is that untransfected cells actually have even more non-specific GABA uptake. We can talk about that maybe at the end. But it doesn't, for the purpose of our assay, it doesn't seem to interfere. We seem to I think we've done, we've done appropriate controls to limit the effects of this at the relevant test concentrations of GABA. But it's an interesting question to come back to. And so again, with this this kinetics sort of difference between these assays, they're very well correlated, but we sort of wondered why. So we discussed this with Mike Kavanaugh, and he suggested we do some time course experiment. And so we'd taken a pathogenic variant, one that is recognized by multiple sources to be pathogenic, but does have some residual function, the V342M, unlike other ones that appear to have almost no function at all. This one, by multiple sources, are reported to have some residual GABA uptake. And we just did separate assays, not the same one over time, but separate assays that were incubated for shorter or longer durations. And we see that there's these curves. And if you fit them to just a very simple pseudo first order association kinetics that relates the plateau to the rate of change, it would appear that there are differences in the steady state IGABA that is achieved by, in this case, a pathogenic variant versus the wild type control, that are mathematically, or at least explained by this this change in the plateau, as opposed to these things could be independent. You could have a slower rate and a slower plateau, or you could have a slower rate and a normal plateau. But it would appear to be the case that the steady state level is altered in these variants. And so then we did another assay that I think we weren't expecting these results. We're not the first people to do this assay, but it's an interesting one. The question arises, what's the actual validity of assessing GAT1 function in what is more or less a homozygous state, because the only GAT1 being expressed is the mutant GAT1 in these cells. And would you see something different, or is it more informative if you were to combine mutant with wild type, which is more reflective of what a cell might be doing in vivo in a patient that has an SLC6A1 related disorder? And what we found was that for the most part, we see exactly what you would expect. And so in this, we did these two experiments in parallel, one in which there's a mutant transfection, and the other where we've got stoichiometrically matched amounts of mutant versus wild type in combination. And for the control for this group, we just used 2x wild type together, so 2 micrograms of the wild type DNA in these cells. And what we found was that for the benign variant, we see that there's an additive increase that didn't reach significance, but does seem to be going up. And then across all of our pathogenic variants, we see what looks like an additive increase. And then for a few of our variants of uncertain significance, in which our IGABA assay had shown reduced levels in the sort of homozygous state, we also see this additive effect. And that's exactly what you would expect, based on other people's data and based on our understanding of the protein's function. But then for a large number of variants that had residual GABA uptake activity, we found that there seemed to be this non-additive effect of adding the wild type. So for example, for these variants, L54F, S574R, actually looks like the amount had gone down. And for other ones, there just seems to be we don't see the expected additive effect. And looking at these in a somewhat cursory way, we don't see any sort of thing that jumps out to us with respect to their position in the protein or their position in a domain. It would appear to be the closest thing that appears to explain it is this level of residual protein function from the homozygous state, with the exception of the fact that the benign variant here seemed to have a different response. And so we have at least one example in which it would seem that the assay has the sufficient dynamic range to see an increase in the GABA uptake from a high residual level in the homozygous state to then the heterozygous state. But for these variants, it doesn't seem to be the case. And so we'll talk a little bit more about that, but I'm curious to get people's thoughts as well. And then, so we were hopeful to be able to use this assay to functionally characterize variants from novel, newly identified variants in patients or VUSs in patients that remained uncharacterized. And so despite over 200 variants having been described in this recent paper by Silva et al., there was a patient who had come to our attention with a variant at M55T that had not been assayed by any other group. And just a brief summary of this patient: developed epilepsy, not genetic generalized, not EMAS, but rather what appeared to be focal onset seizures at the age of 4, has ASD, has moderate to severe intellectual disability, has a normal MRI, and other genetic testing was negative, leading up to a whole exome sequencing that showed a VUS in SLC6A1, this M55T, and in truth also had a VUS in GRIC2. And so this patient was a little unclear, but it highlights the challenges that are faced, I think, every day by clinicians who are trying to figure out what the explanation for our patients' seizures is. But suffice to say, it was believed that SLC6A1 could account for this patient's symptoms clinically. And so we had generated this variant M55T in our assay. I should mention that the pathogenicity prediction scores are mixed. Overall, it's predicted pathogenic by the variant prediction algorithms, but some predicted to be benign, notably polyphen 2 and metal R. And we ran this in our assay. And we were surprised to see that there's absolutely no difference. And we ran it a couple of times, and there seems to be no change in the GABA uptake. And just to put this in the context of the polyphen 2 score, it seems to be there seems to be, there's a good correlation I didn't tell you about between our results and polyphen 2 score, and M55T seems to be sitting here in that distribution. And again, we don't know what to make about this either. We'll talk about that maybe in some of the later slides. There's, maybe trivial explanations for why this patient may appear this way, but there is sort of a broader question related to variants that by GABA uptake assay appear to have no effect. And what we should do about that, what do we think about that, and whether we're convinced by an assay like this that a negative result is proof that it's not pathogenic. So then we went on, as I mentioned, we were interested in developing this thing for drug screening. So we did evaluate the effects of 4-phenylbutyrate shown here. And for this, we used our stable cell line. We used I think in this case, we actually used a 96 well format, but we've also done it. We've done evaluations in the 3 to 4 well format using automated tools as well. And in this assay, we do see an effect of 4-phenylbutyrate, which again is expected and what you would like to see. But it is notably, I would say, assay condition dependent. So at our 1 micromolar GABA, which is the condition that I've been describing as probably being the most, it's in the sweet spot in terms of the magnitude of its effect and its specificity. We do see an effect of 1 millimolar and 3 millimolar 4-phenylbutyrate with respect to an increase in the IGABA that's measured. And this is roughly in line with published data using similar concentrations over longer periods of time, like 24 hours in some cases, and this being in 6. But what we did notice was that at the lower concentrations of GABA, I mean, you might expect one of the effects of an agonist or a molecular chaperone would be that it could increase the levels of GABA uptake at even lower affinities. It doesn't seem to, maybe there's a trend, but it not a significant effect at the lower GABA. And then at higher GABA, although for one of our vehicle controls, it appears to be different. For other vehicle controls like DMSO, we see that we actually have a similar level of a difference in the GABA uptake. So it appears to maybe be assay dependent condition, but that's the reason I suppose that you optimize for specific assays or specific conditions and not others. So the long and the short of it is that we have a positive control. It seems that our assay using IGABA and not tritiated GABA does seem to recapitulate effects that have previously been seen for 4-phenylbutyrate, which increases confidence that the assay could be used for further drug screening. And so we have not yet we've not yet executed a drug screen, but we have reduced to practice some of the elements that would be needed using automation, using 3 to 4 well format, and just to show some of the benchmarks that we had developed. We'd performed some. We'd assessed 2 different ways of evaluating the data: raw IGABA versus an adjusted IGABA that uses a linear model to account for the change in the position of the well on the plate. These are live cells. We can't kill them, so in theory, the activity of the GABA uptake, although it's meant to be at a semi steady state, could be changing over the course of the plate. We've shown that doesn't appear to be a major factor, but in addition to that, we've performed some bootstrap simulations just to ask, what would the thresholds be for defining some hit above background, and what would be our ceiling for this assay based on a positive control. We didn't have 4 PBA at the time, so we had just used GABA at a higher concentration, still a specific one of GABA 5 micromolar, to ask this question. And without any, sort of naive approach, without any fooling around with the way that the data is normalized, it's something of a narrow window above the background with respect to, identifying hits. But with some normalization, we found that we can expand the range. And this is essentially assuming a screen in which one concentration of a library is tested against different numbers of controls, negative or positive controls on the plate. And with this format, we think we have a wide enough window to possibly do some compound screening with this assay. And so that's everything I have for you. And in summary, we think that this assay is suitable for assessing deficits in the whole cell GABA uptake, seems to be able to assess pathogenicity of coding variants, highly correlated with radioactive tritiated GABA. We see that a mild pathogenic GAT1 variant seems to alter the steady state of IGABA. This novel M55T variant is not associated with altered IGABA, and that the use of a stable cell line could be useful for compound screening to identify agonists of GAT1 using high content imaging. And so this brings me to one of the questions that had sort of come up by surprise. We were really just looking for an assay to use, but the question of these mild loss of function variants in SLC 681. So for our study, they sort of came up in two places. One was this case in which there's just no change in the IGABA, but the other one was in the studies in which we had combined the mutants with residual GABA function with wild type, and seen that the level had gone down. And so it's important to observe that apparently these mild loss of function variants, you define in different ways, are very common. So based on this Silva et al. Paper, 40% of the variants that they had reported seemed to be in the typical GABA uptake range, which based on their benchmarks was between a measured reduction of 49.2% or a measured increase of 47.5%. So it's nearly half of the variants that are being reported based merely on GABA uptake would sort of be, classified as potentially unrelated, is what the authors, I believe, were concluding in some cases. So in addition to that, 14.6% are really unchanged or hyperfunctioning. So half of that range, maybe corresponding to like the variant that we saw with the M55T. So again, these aren't rare, and with respect to them having arisen amongst our patients. And so one of the questions is, are we confident the variants that have normal GABA uptake by these cell-based assays are incompatible with pathogenicity? What do we think of variants with mild deficits that show non-additive effects with the wild type GAT1? Possible interpretations that I'll put out there just to start the conversation. Maybe these effects are true, they're real, and there's some other disease mechanism. Surface expression wouldn't seem to account for it. I mean, it's a well-described phenomenon, but these are, might account for severe function or severe loss of function that had residual GABA uptake. This assay is sort of inclusive of that. Like if there was a reduction in surface expression, I would expect that result to be reflected in our results as well. Maybe other things that I don't know as much about, like oligomerization or interaction with specific proteins, which I think I haven't seen many reports about, could be related. But I would observe that this could have implications for variant interpretation and also patient eligibility for genetic therapy. If we think that the variant isn't the cause, but it really is, these patients could go untreated. Or another possible interpretation is that it's misleading. Maybe these are artifacts of these heterologous assays. It's somewhat artificial to be putting these cells, putting these proteins in these cells where they don't belong, and to expect that they're fully recapitulating all the functions. Maybe it's true for a large proportion of them, but some of these subtler variants, maybe there's something more to it. Maybe we should be assessing them in more neural cell types, which authors have done, or in vivo animal models. And with that, I'd just like to acknowledge the members of my new lab at Northwestern, as well as former members of my research group at Boston Children's, and particularly Guochi, who did a lot of these cell-based assays, and Paduri, my mentor, Kim Wiltru, who had submitted information about this patient, and very helpful conversations with Henry Lee, Jed Hubbs, Michael Kavanaugh, and as well as Lee Barrett, who helped with a lot of the initial setup of the assay using the IXM. Thanks.
Okay, does anybody have any questions?
Hi, Chris. That's really nice. This is Patty. I have a question about the non-additive effect from the wild-type allele. So in our mouse model, we studied Maxwell's mouse model S295L and A288V. So 288V has about 35% remaining effect, but Maxwell almost has no effect. However, in the mouse model, yeah, so I was going to share the paper with the group. I can put in the chat. So in the mouse model, you with the tigabin binding, you almost see no additive effect. So actually, we almost see identical effect. So in SL and AV. So that means the 35% were identified when you express the Newton alone, you will not see in mouse model. So that's kind of what you identified here. I also have a question about how much cDNA you use. You mentioned 2 microgram.
Correct.
You transfect it into cells, right?
Correct. The format for this was I would have the exact plate type that we were using. I'd have to review, but the format was typically that we would perform the transfection actually in a plate, but then reseed the cells into a 96 well plate. So yeah, the amount of DNA might be higher than is seen by other groups, but the number of cells involved was possibly higher than what are used in other assays as well.
How big is your plate when you transfect?
I think it was 10 centimeters.
Okay. So that's the DNA amount is not much. If it's 10.
And to your, well, sorry.
Go ahead. Yeah, continue. You go ahead.
Oh, I was going to say, yeah, we were, you know, in writing up this manuscript in preparation, we had been reviewing, right. One interpretation of the non-additive effect is essentially like a dominant negative effect, like somehow it's interfering with wild type GAT1. We reviewed the literature for examples of this, and yours came up exactly what you said. That might be actually the strongest example in the literature so far that would suggest a dominant negative effect. But I felt like I had heard at meetings, that people had seen evidence of other ones, but then when I went looking for in the literature, I couldn't find them. So I mean, for example, at some point I thought I had been told that the S295L variant might have a dominant negative effect, but then when I went back to look at these papers, it appears it appears to be loss of function in the same way as other ones. So yours stood out as maybe being the one in vivo piece of evidence that suggests that yes, there could be some evidence of a negative interaction there. Interestingly, though, for the A288V allele, which is where you saw sort of this, there was greater residual activity, right, in the homozygous A288V versus the S295L. The A288V in our assay has no function. So it's like we saw an additive, I mean, at least within the confines of our assays, we saw an additive effect when we combined wild type with the A288V. It was these other variants that had more residual function that seemed to have the non-additive effect. So it could be assay dependent, but I agree that it sort of corroborates that there could be something more going on.
Did you see additive effect in Maxwell's S295L?
Did we? Yeah, we did. Yeah. And that was, yeah, we had been expecting I think we had heard that the S295L might be dominant negative. And so initially we were, that was going to be our bellwether or whether that assay was working, and it had an additive effect. But when I went through the literature, I had trouble confirming like the evidence that was suggesting non-additive effects or, dominant negative interactions with the S295L.
So you don't think it's a dominant negative?
All I can speak for is my assay conditions, and it doesn't seem to demonstrate that non-additive effect. We see a nice additive effect of combining it with wild type. And when we looked through the literature, the published data that I had seen, I believe, was related to seizure activity. Let's see, Lindquist 2023 had compared like a 295L mouse versus a knockout mouse. And although there were differences in the mouse strains, they had reported that there were similar rates of spike wave discharges. And the interpretation was that they were maybe similarly affected. Whereas you would have expected a S295L het to be more significantly affected if it was a dominant negative. But again, I'm appealing to the room to educate me on this. I can only report what I've found from these recent reports.
It is very variant dependent. Also, that's I think the window. If you say co-express wild type and the mutant, if you see effect or you do not see, it's very like a narrow window. We test if you give a little more cDNA and then you lose the effect. So you have to maintain, like for example, if we use 0.5 microgram, you will see very robust effect. But if you increase to 1 microgram in total, you don't need you don't have the effect anymore. So I think in vitro system, probably it's very dosage sensitive, and but in mouse model, in mouse model, it's very consistent. A2A the protein is more stable than Maxwell's. So Maxwell's in homozygous, you almost have no mutant protein detected, but in A2A you always have some from a homozygous animal. And then, but in the heterozygous, as you said, you may have a negative interaction between the wild type allele and the mutant allele. So when actually the data was nicely provided by Roche for us. So if you use TNF binding, which will probe the total GAT1 intracellular and extracellular. So they are shocked because there is no difference between two mouse models, and which is quite similar to what we see if you use different tag for wild type and mutant allele. So the additive effect is not what you see in homozygous, is what you see like with the interaction between wild type and mutant. So it's more complicated in the heterozygous condition.
Chris, there's a lot of comments, and it might make more sense if you just skim them real quick.
Sure. Yeah, let's see. One question from Moritz: do you have separate fluorescent tags on each variant for the heterozygous expression that you can know the relative surface? So in the assays that I showed, the answer is no. But each variant, so the variants that we have this P2A linker, so it's not a fused protein. But in data I didn't show, we have run validation assays for the wild type fusion protein, and it performs similarly well. And each of these variants could be converted into a fluorescent variant just by deleting the P2A to show that the surface expression was affected or unaffected. But we didn't look at that in this particular assay. Sarah asked, have we tried the assay in neurons or astrocytes? We have not. I believe there's descriptions of it in the literature, but we hadn't tested it in this setting. Another question was, can it be run in patient cells? Yes, it could be. We haven't tried that, but no barrier to doing that. Let's see. And maybe some of this negative data has to do with the overexpression. Yeah, I mean, my thoughts are that there could be there could be some features of these heterologous assays, be them, maybe there's cell-specific factors that we're missing, and if we put it in there into a patient cell, or to human iPSC-derived neurons, or just other cell lines that were more resembling neurons or astrocytes, maybe we would see something different. So yeah, I would highlight that maybe as a next step that would be of great interest. Michael Rogowski, good to see you. Can you assay an organized tissue that exhibits seizures such as slice organoids? How about simultaneously assaying extracellular GABA? Yes. So I'm not the inventor of the iGABA sniffer. I think maybe Jonathan Marvin, who's also on this call and has another question coming up, could be rightly considered one of the originators of this tool. But my understanding is that yes, it has been used. There's examples of it being used in intact tissue in organisms. Organoids, I'm not sure, but there shouldn't be any barrier to that. And then simultaneously assaying extracellular GABA. Simultaneously assaying extracellular GABA, I think you might have to choose. I mean, but the way that it's been described in the literature, we actually modified the protein that had been used in vivo to show extracellular GABA. That protein has some sequences that allow the iGABA sniffer to be trafficked to the cell membrane on the extracellular side, and can be used for that. And then Jonathan says, screen libraries, have we validated with inhibitors? Yes, we have. So actually, I didn't include any of that data. Some of the before we even dove into all of these things, we absolutely showed that tiagabine blocks the specific effects in these cell lines. And in fact, in the optimized protocol that we're using here, we also noted that basically pre-treatment with tiagabine, like overnight, followed by a wash before we start the assay, improved the signal to noise. And so we are actually using tiagabine early in this assay to almost like as a way of starting the assay on the day in which we do the imaging. Great.
Hi, Chris. This is Henry.
Hey, Henry.
Good to see you here. May I ask
Yeah,
you a
good to
question?
see you.
Good to see you. This is great progress, and congratulations on all the very interesting results. I want to
Thank
make
you.
a quick. When I see the additive effect, I think it's that very encouraging. That essentially you're saying that with the wild type presence in the combination of the mutant, you actually can see a rescue, right? And I think for some of those variants. My question is, do you see a peak of that? How far you can get to the rescue? Like you're using 1:1 ratio, if I understand correctly, in your additive assay. And I don't know if it's worth to try a different ratio of those wild type 2 variant to see how high you can get back to that. Is there any maybe some utility or some meaningful, some information on that? And also thinking about some of the other variants that has the residual level, then you don't see additive effect. Does that mean that maybe gene therapy will not work on those? That could be something we need to think about. I think that's my take on that. But I think this is very, very interesting and informative, essentially.
Great. Thank you. Yeah, thanks for those thoughts. Yes, for the ratio, I agree. I think that's a good. If we were to go back and do more analyses, I think changing the total amount that we're using in this transfection and assessing a different ratio would be a reasonable thing. Yeah, it would at least give us a sense of maybe a dose response, or if there's some nonlinear effect, which would be easy to do and to see. And yeah, I don't want to. I'm not going to pull the alarm bell and say that it's definitely the case that these phenomenon exist in a way that will be clinically meaningful without doing some more investigation. But it does, you know, if it is a phenomenon that exists in vivo or is held up by other assays, it would suggest that, yeah, you might need to do some more thought about merely adding another gene, you know, on top of a certain mutant gene, if there really is an interaction there. Jack Parent asked about the 6-hour time course. Yeah, so I think there's a slide I didn't include. We had shown, I wonder if I can just pull it up. There's a steady state in the GABA. So we tried shorter time periods. Oh, I'm sorry. You're asking about maybe the one with the variants, infusing the GABA. No, the GABA is just exogenously applied at the beginning of that 6-hour period.
Yeah. So,
And
why does it take 6 hours to peak?
Yeah, that's a great question. I mean, that was one of the original, when we first started this assay, one of the things that was slow or that slowed us down with its development was like this, I would say, unexpected slowness with respect to the accumulation of the fluorescence. I mean, I think that if we could actually measure GABA, we would see that, you know, if you were using radioactive GABA, there's a significant amount of GABA that's coming in during the earlier time points, but the sensitivity of the sensor might not reflect some of the earlier changes as well as the later changes. Would that, I mean, does that speak to your question? Or?
Yeah. We actually, my colleague, you know, tried it once, but only waited 10 minutes, and
Right.
that wasn't long enough.
Exactly. Yeah, we were surprised at how long it took, but I think based on its sensitivity, it just requires more to accumulate. It also speaks to the, you know, some of our trepidation about comparing the results of this assay versus treated GABA before we directly compared them, you know, by correlation. One of my concerns was that, are we really measuring the same thing because we're giving it 6 hours and it's all going in there, reaching a steady state? The math would suggest that they seem to be very well correlated, and so they are likely features that are linked. But yes, it would, to us, it was also a surprising length of time.
Thank you. Very nice talk.
Thanks.
Hey, this is Jonathan. I just wanted to comment on that aspect of the concentration that you need, and that is one of the limitations of the F102Y Y137L variant of the sensor, is that while it does have the highest change in fluorescence, its affinity is, you know, significantly weaker than some of the other variants. And, you know, we have not been able to create a variant with a high change in fluorescence and high affinity, but, you know, I think that would resolve the issues of the time course that it takes. Because yes, you have to accumulate a certain amount of GABA in the cell before you can even detect it.
Gotcha.
That makes sense. Thanks.
And yeah, so for us, I mean, 6 hours, it's not too bad. If you're staggering these assays, it's sort of just all hands off time. But I suppose in the long run, faster assays are always better. We have not tried the iGABA sniffer 2, but I think maybe to what Johnny, I'm not sure how that one relates to what Johnny was mentioning, but yes, I think maybe a higher affinity one.
So I'm just gonna say the problem with that one is its total change in fluorescence is actually lower than the original iGABA sniffer, but it has significantly higher affinity. So it's great for, well, it's good for observing extracellular GABA, you know, with regular neurotransmission. I don't think it would be the best variant for your purposes.
Gotcha.
Yeah.
And then Caddy says, are you able to separate the fluorescence produced by the wild type versus the mutant allele in the heterozygous? Not with this particular tool. So in this case, we've got, you know, the iGABA sniffers responding to GABA from all sources. And so in the cells that have the combined wild type and variant together, they're sort of both letting GABA in, and then the sensor is detecting them. If I can't think of a way. Maybe maybe somebody on the call might have some suggestions for this one, but I don't think that we have the ability to separate, GABA from one versus the other when they're in the combined state.
Thank you.
All right, this is great. Does anybody
All
else have
right.
any questions? Well, thank you so much, Chris, and I will
Thank
send
you.
out a recording to everybody and along with your contact info for follow-up.
Great.
Have a great day, everyone.
Thanks, everybody.
I have a question.